So we'll kick off now with Gilles Atlan from NF2 Biosolutions UK and Europe.
So thank you for coming. My name is Gilles Atlan. So I would like to dedicate this presentation to Alan. Unfortunately, last week he passed away from NF2. Alan was a great supporter of research for NF2, and he lost this fight. And that's one of the reasons why I'm here today, for people like Alan, but also for my daughter. I'm the dad of Karen Atlan. She's 15 years old. She's diagnosed with NF2 since we did the diagnosis when she was 6 years old. And so Karen, my daughter, she had already 2 surgeries of her vestibular schwannoma. We were lucky it went well, but obviously, like every NF2 patient, she has many more other tumors to take care of. So she's the reason why 4 years ago, we started NF2 BioSolutions with a group of patients and NF2 families like Claire, and under the leadership of Nicole Henwood, that she's an anesthesiologist, MD, and she has a son with NF2. His name is AJ. We started this nonprofit with the goal of looking at what are all these novel therapies for other rare disease and see if it could be applied on NF2. In the last few years, we successfully fundraised up to $2 million, and with it, we could funded 12 different labs with different approach and for different goal. And all this has been possible thanks to all the patients, family, and friends. That's the source of the funds that we mostly received. So that's why it's very important that we will be all together in it in order to really make a change for this disease. So these are the different projects that we jump started and supporting during the last 3-4 years. So why there is different project? Why we don't put all our efforts on one project? Simply because there is different goals. One goal could be destroying existing tumor. Another goal for the patients could be stop the tumor that they have to grow. And another goal could be that we don't want any new tumor to appear. So there's different goals, so there should be different approach as well. And also we don't know which approach is going to work at the end. So it's better to start in parallel many different approach and maybe at one point of time combine them or move forward with the one that has the best results. So one of the approach is the suicide gene therapy, where basically we add a gene into the tumor that create the death of the tumor cells. So using a virus to deliver that gene. There is already good results on mice, and there is a biotech that is trying to move it forward to the clinic. I have just 10 minutes, so I'm going to focus only on the bacterial immunotherapy and on the gene addition therapy. But in a nutshell, about the others, we have the anti-inflammatory project that Adam and Grace presented today, and they are going to present it again tomorrow as well. That is in Manchester. And the TIL immunotherapy is a project that we are going to start this year or beginning of next year here in Manchester as well. So let's talk about the bacterial therapy. Okay, so the first one of the lab that is the most advanced currently has a very interesting approach, is to inject a Salmonella type of bacteria, genetically modified, into the tumor, and it starts to really decrease the size of the tumor. It seems like it's affecting the immune response, and the immune cells are starting to attack the tumor. So you can see in the graph at the week number 2, they were injecting the bacteria, and the control, it means the mice that didn't have this bacteria injected is the black one, continue to grow, the tumor continues to grow, but when we injected with the bacteria, you see the green and the red, it today decreased almost to 0. So that's very encouraging. Currently, the researchers are developing a new strain of bacteria that will be armed with another gene also to accelerate the destruction of the tumor. The challenge is that it takes a long time to create this new strain, but that's why in the meantime, in parallel, having a biotech that is preparing the trial for this research. The name of the biotech is Mulberry. They successfully raised already $2 million plus a grant. And now they are preparing a trial for this research that hopefully will start in 2023 if we can raise the funds. So that's quite well advanced, but it is at the phase where a lot of funding are needed. Next slide, please. Next project I would like to talk about is the gene addition therapy. So the gene addition therapy, we started it at Nationwide Children's Hospital in the lab of Dr. Meyer. It's a lab that has a lot of experience in gene therapy. They successfully bring to the clinic other gene therapy for other rare disease like SMA, Batten disease. We jump-started this project from the beginning more than 2 years ago. And the idea here is to add a healthy NF2 gene. So the healthy NF2 gene can produce the protein myelin that is missing. And that protein myelin is making that the tumor is not growing anymore. So what Dr. Meyer has been doing during the last 2 years, she created a new cells line derived from patient skin. So she transformed the cell skin into Schwann cells. Thanks to these techniques, she developed very quickly 3 different types of cell line. She also created a second hit, so she turned these cells into tumor cells as well. And that's very important to have this cells culture, otherwise you are going to have a hard time to develop your approach. And then she tested on this cells line a construct using a virus to affect the cells. And it successfully increased the level of myelin and reduced the cell growth. We are starting now to have some good results in mice model, also increasing the expression of myelin, of the protein that is missing. And in another type of mice model that have a xenograft tumor, we see a significant decrease in growth, in tumor growth. So that's very encouraging to have these good results in mice. And we are at the point where we can also start to do some work to prepare, to get ready for a trial. But that's the big investment that is needed at that point of time. And the issue is that there is a funding gap, basically, that is happening right after the preclinical work. As a foundation, as a nonprofit, we can fundraise enough usually to do the work of the pilot and the preclinical work. But to prepare for a trial, all the application that is needed for the FDA and to recruit and do the phase 0 and phase 1, it costs a lot of money. And usually doing a phase 0 and phase 1 is still very risky for private funds, for biotechs and for pharma. So they are willing more to invest when we are already in phase 2. So there is a gap here of funding, and trying to resolve that gap, we partner with Vibe, another nonprofit that identified this issue of funding the first phase of a trial. And their goal is really to help some nonprofit companies to be able to fundraise the funds that are needed for doing the first trial. And we partner with them, created a biotech myelin therapeutic, and right now we are in the process of raising the funds so we could move to the previous research that you have seen, to move it to the trial. And there is not only approaches we need also tools for these labs. These labs can progress, or they can test their approach if they have the right tools. So we also funding NF2 Biosolutions, thanks to the donations of all the patients' families. We are funding also the creation of some tools. One of the important tools is NF2 Biobank. And the goal of the NF2 Biobank is to collect enough tissues from patients, blood, saliva, and also with some fresh tissue, we can also develop cell culture as well. So we worked first with a Children Brain Tumor Network from Children's Hospital of Philadelphia. We finance the creation of this NF2 Biobank. So they did great work at collecting tissues, blood, saliva. The challenge was after that to share them to other entities. It's a complex process. So in order to speed up the sharing of this culture, of these tissues, we are now in the process of transferring the ownership of these biosamples to us, to NF2 BioSolutions. So we will be able to share the tissues and the cell lines in a efficient way. One thing that we did with the CBTN was to collect 50 trios of saliva. Trios meaning the kids with NF2 and the DNA of the parents. So it means 150 samples we collected, and with these samples we are doing the full sequencing, genome sequencing of these samples. Currently, the raw data are being received, and it will be displayed online through the NIH Kids First program. So all the researchers around the world will have access to full genome sequencing of 150 people, 50 trials of NF2 families. The second tool that we funded, we have also the NF2 epidemioma mice model that Dr. Calamaridis is going to present tomorrow, because there is no mice that are developing NF2 epidemioma. So if we create if there is a new approach, a new construct and we want it to be tested for epidemioma, there is no abilities to do it on the mice. So that's something that was really missing, and now Professor Calamaridis is working on creating that mice. And the third thing is the need of having an automation to automatically measure the volume of vestibular schwannoma. Next slide, please. So the problem that currently we have is that usually when you are doing an MRI, in the report, they give you the measurement of your tumor in 2 dimensions, just X and Y. Or sometimes in 3 dimensions, but there's 3 measurements, just 1 X, 1 Y, 1 Z. But the problem, that's not the real size of the tumor. The tumor is not a ball. So you need really a more accurate type of volumetric measurement. And the goal of this project is to automate, and it's possible to do it, but manually it takes a lot of time. The person has to go through every slide and measure and then do the whole calculation of the volume. So we want to automate this volume measurement. And so the patients and the researcher, because we will have also to calculate the volume of the patient when we do trial, could have a way more accurate the measurement of their vestibular schwannoma. So this is an example, for example, of a patient that on the left side, you can see a linear measurement, usually the measurement that you are getting on your MRI. You can see that from 2016 to 2017, it even showed that the tumor decreased, or from 2019 and 2021, it showed roughly stable. But in fact, between 2016 to 2017, it grew by 33% when the patient thought it decreased. Or here, between 2019 and 2021, it grew by 20% when he thought it's relatively stable. So it's very hard for a patient and for the clinicians to take decisions, to take a risk maybe of doing a surgery or starting Avastin or drugs if you don't have the accurate measurement. So that's why we developed with Yale, with the lab of Dr. Visna at Yale, the ability to automatically do an accurate measurement of the tumor, but also to visualize it in 3D. For example, that's the vestibular schwannoma tumor. The red part is the tumor in 2021. The yellow part is the part that grew from the tumor 1 year later. And in that example, the tumor grew by 50%. So ideally, more patients will have access to that to take better decisions, but also researchers will have access to that so they can test, have a better way to do the measurement during the trial. Next slide. So all this happened, all this research that I talked about happened thanks to all the NF2 patients and family that donated to us. We tried, obviously, to get donation from other sources as well. I know in UK, you are getting quite some success with some grants, with some other association. In US, it's a bit more challenging, but people, families and the patients and the friends are contributing a lot with their donation. And you can see that the donor segments, most of the dollar donation and the number of donation came from NF2 parents, family, and friends. So that's why, and all this has been possible thanks to the NF2 community. So we always need help, especially also in UK. If you would like to volunteer or to donate, that will be very helpful so we can continue to do all the work that we are doing currently, and also support other new approach, because there is a lot of other novel therapies that have not been tested, and also moving the current project more toward the clinic. Okay. So thank you very much. And again, it's all about the network, connecting the patients with the biotechs, with an NF2 expert, with the lab, in order for making a big difference in the research for NF2. Thank you.
So we just have a question here.
Actually, more than one. I'd like to ask you, you were talking about viruses, bacteria, and so on, how they are delivered to the tumors.
So depending on
The second, please.
It needs to reply using the microphone.
So depending on the approach, in the case of the bacterial therapy, most likely it will be intratumoral. So yes, intratumoral, or could be in the CNS, in the central nervous system. That's depend, and that's the thing that will be tested during trial.
Apply an injection. How to apply an injection in a tumor? That they are often very small and so on. And I can now make the third question, and it's the last. How about RNA to produce myelin?
So regarding the injection, it's possible to inject inside tumor. So it's being done for other diseases as well, but it's accessible. And even the vestibular schwannoma, it's possible to do a small holes in the skull. We talked to a neurosurgeon, and that's also feasible.